Hi everyone, I had this idea to make a little follow-up video on my latest video on LAMP. If you haven't checked that one out, I strongly recommend you head there first. I explain everything that is to know about LAMP, what it is, how it works, and also some examples of how LAMP is used in diagnostics. I will link that video in the video description and also here for you. In today's video, I will do a direct comparison between LAMP and the current gold standard of PCR. I will talk about similarities, but also some areas where these techniques differ from one another. And in the end, I will speak about advantages and also disadvantages of both techniques. Okay, so let's get started. If we look at the number of enzymes that is required for a PCR reaction, this is typically just one, and for LAMP, it's also just one. There are some isothermal amplification techniques that use multiple enzymes, but for LAMP, usually it's just one enzyme. The primer design and the number of primers in PCR, the design is considered a simple design, and we typically, for a conventional PCR, only need 2 primers. That need to work with each other. For LAMP, this is a bit more complex. Here we have 4 to 6 different primers that need to properly work with each other. And that's also why the design of LAMP primers is considered a bit more complex. There is software that can help you to do that. And I'm also going to link one here in the video description, which is Primer Explorer. This is a browser-based tool that can help you to design LAMP primers, and it works very, very well. The temperature in PCR, we have thermal cycling at different temperatures. This is required for our template to be amplified and for the whole reaction to work. In LAMP, this is actually isothermal, so amplification at a constant temperature. And typically in LAMP, this is around 60 to 65 degrees. So that's a very big difference between PCR and LAMP, right? One needs thermal cycling, the other just needs one constant temperature. Reaction time. The reaction time in PCR is usually something around 1.5 hours or even longer. This also depends if you have a reverse transcription step before your PCR. And in LAMP, this is usually below an hour. And actually, many LAMP assays have much, much lower runtime. So sometimes 30 minutes, 45 minutes, but mostly below an hour. So it has faster turnaround times than PCR. The target in PCR is DNA, as well as in LAMP, but you also can have RNA targets, for example. Here, of course, we need a reverse transcription enzyme, and this we can also add to our LAMP reaction, so there are reverse transcription LAMP assays available as well. The amplicon in PCR is actually a nice double-stranded DNA amplicon, but in LAMP we have these concatenated DNA amplicons, which are basically a series of repeats of different sizes, and this can make the work with those amplicons a little bit more tricky in LAMP compared to PCR. Detection methods in PCR and LAMP, they both share similar detection methods, for example, gel electrophoresis or real-time. In PCR, we also have some ELISA-based options available. But in LAMP, what's really, really cool is that we can do here a colorimetric readout, meaning we don't need any device at all to read our sample. We can actually use our naked eye. And this is what makes LAMP also so attractive for point-of-need diagnostic applications. If we look at tolerance to contaminants, PCR is considered to have a low tolerance towards contaminants or inhibitory substances. These can be found in multiple types of samples. It's very typical for environmental samples, for example, and LAMP is considered to have a relatively high tolerance towards these inhibitory substances, which is very nice. The initial template denaturation in PCR, yes, we need that. In LAMP, no, we don't need that. We don't have that. We can just heat up to our constant temperature. And this is all we need to do the reaction. Template processivity in PCR, this is clearly heat. We need to heat up and cool down to different temperatures, and we need to do this over and over again in cycles to facilitate our amplification reaction. In LAMP, we actually have the strand displacement property of the polymerase, meaning that the polymerase can actually do the job itself of opening up the double strand and performing amplification, and that's why we just need one constant temperature in LAMP. So at the end, I also wanted to talk about some advantages as well as disadvantages of PCR and LAMP. And when we look at PCR and its advantages, it is a very, very accurate method, and it's also very robust in its quantification. It's fairly simple, and another thing is that it's very, very well established. So I would say that almost any lab in the world can perform a PCR these days, And that also leads to a lot of knowledge that is available for PCR. So whenever you have a troubleshooting or you want to establish a new assay, there are tons of information on the internet that you can use to perform your assay in your laboratory as well. And also we have a lot of commercial kits available. I would say for any pathogen or any application that you can think of, there's usually a PCR assay already available that you can buy online. What are the advantages of LAMP? LAMP is considered to really yield a high amount of amplification product in a short time. That's really crazy with LAMP. This can be an advantage, but it can also be a huge disadvantage if you think of things like contamination and stuff. But usually LAMP produces a lot of amplification product. Again, LAMP is considered to be very tolerant towards inhibitors, which is nice, and it's also highly specific. If you think about the number of primers that we use in a LAMP reaction, 4 to 6 primers, this makes LAMP very, very specific. It also allows for the detection by naked eye. Again, this is the colorimetric readout that is available with LAMP, and that opens a lot of possibilities for point-of-need diagnostic purposes. That's really cool. And also now we have some commercial kits available for LAMP as well. And I guess in the next years, this will become more and more and more. Let's also talk about some of the disadvantages. In PCR, we have relatively high equipment cost. A PCR cycler itself is not so expensive, but for example, if we think of a real-time PCR cycler, these can become fairly expensive and they're also fairly sophisticated equipment. The reaction can be easily inhibited by contaminants in the reaction, and also inhibitory substances, of course. And also we have these fairly long amplification times, especially if we compare this now with the isothermal amplification. What are some disadvantages of LAMP? It is actually not suitable for small targets. As you can imagine, if you have to fit 4 to 6 different primers in your target area, this is not going to work if you have a very, very small target that you're looking for. It is also described that LAMP is prone to false positive results. This is also because these primers need to somehow work with each other. And if you have primer-dimer formation, this can lead to false positive results. So when designing LAMP primers, it's very, very important to check whether these primers are actually specifically very specific in your reaction. And also, we have this fairly complex primer design. Again, yes, it is more complex than in a conventional PCR, but there are tools that can help you, for example, Primer Explorer. And again, I'm going to link this down in the video description. So this was the direct comparison between LAMP and PCR. But the question remains, will LAMP take over and become the new gold standard in the future, or will PCR keep that title? Let me know what you think in the comments. Also, I would really appreciate if you like and subscribe to my channel. Bye.